Today, we are back with another video on chromosome structural abnormalities. So, let's get started. Well, chromosome abnormalities are a principal cause of pregnancy loss, an estimated 10 to 15% of conceptions having a chromosome abnormality, of which 95% are lost before term. Close to 90% of the abnormalities are aneuploidies. What is aneuploidy? Aneuploidy means having missing or extra chromosomes. The risk of having a child with an aneuploidy increases as a woman ages. Trisomy is the most common aneuploidy. In trisomy, there is an extra chromosome. Around 2/3 of the structural abnormalities arise in oocytes, 1/3 in sperm, the latter increasing with paternal age. Structural aberrations include translocations, deletions, ring, duplications, inversions, isochromosomes, centric fragments, and fragile sites. Most of these result from unequal exchange between homologous repeated sequences on the same or different chromosomes, or when two chromosome breaks occur close together, and enzymic repair mechanisms link the wrong ends. Now let's discuss translocations in detail. A translocation involves transposition of chromosome material, usually between chromosomes. 3 types are recognized, which are Centric Fusion or Robertsonian, Reciprocal, and Insertional. Let's discuss them one by one. First type of translocation is Centric Fusion or Robertsonian translocation. Centric fusion arises from breaks at or near the centromeres of 2 chromosomes, followed by their fusion. Let's understand it. This is chromosome 14, and this is chromosome 21, as we have discussed that. Centric fusion or Robertsonian translocation between chromosomes 14 and 21 arises from breaks at or near the centromeres of 2 chromosomes. Then there is fusion of chromosome 14 and chromosome 21. So this is called as derivative chromosome or Robertsonian chromosome. So this is centric fusion or Robertsonian translocation between chromosome 21 and chromosome 14. Moving into details of centric fusion or Robertsonian translocation. The long arms of chromosomes 13, 14, 21, and 22 only are involved, especially 13 with 14, and 14 with 21. These are all acrocentric chromosomes with very small short arms, the latter carrying multiple copies of the ribosomal RNA genes. Their tendency to undergo centric fusion possibly relates to their joint contribution to the function of the nucleolus. Although centric fusion involves loss of ribosomal RNA genes, sufficient intact copies remain on other chromosomes for no serious consequence to result. The carrier of a pair of centrically fused chromosomes may therefore have only 45 chromosomes, but be quite healthy as the overall loss is insignificant. This is a balanced translocation. However, such balanced translocation carriers run into problems at meiosis. Meiosis is a type of cell division in sexually reproducing organisms. That reduces the number of chromosomes in gametes, which are the sex cells, or egg and sperm, with the result that a woman could have many miscarriages. And individuals of either sex can have offspring with effective T21. It is a medical term for having an extra copy of a chromosome, called as trisomy. And Down syndrome. Down syndrome is a condition in which a person has an extra chromosome. Chromosomes are small packages of genes in the body. They determine how a baby's body forms and functions as it grows during pregnancy and after birth. Typically, a baby is born with 46 chromosomes, as we have discussed that. A translocation involves transposition of chromosome material, usually between chromosomes. 3 types are recognized: Centric Fusion, or Robertsonian, Reciprocal, and Insertional. Now let's discuss reciprocal translocations. Well, reciprocal translocation involves intrachromosomal exchange. Let's understand it. This is chromosome 3, and this is chromosome 21, as we have said that. Reciprocal translocation involves intrachromosomal exchange. So, there is an intrachromosomal exchange between chromosome 3 and chromosome 21. Derivative chromosome 3 is formed as a result of binding the long side of chromosome 3 and short side of chromosome 21, whereas derivative chromosome 21 is formed as a result of binding the short side of chromosome 3 and long side of chromosome 21. Either arm of any chromosome can be involved, and carriers are usually healthy. The medical significance is therefore usually for future generations, as carriers can produce chromosomally unbalanced fetuses. X-linked recessive disease can arise in heterozygous females as a consequence of X autosome translocation. Reciprocal translocations can also activate genes in cancers, as in Burkitt lymphoma. So, let's understand. Translocation causing Burkitt lymphoma. This is chromosome 8, with cellular myelocytomatosis. And this is chromosome 14, with immunoglobulin C chain. Now, an intrachromosomal exchange between chromosome 8 and chromosome 14 occurs. This is a part of chromosome 8, and this a part of chromosome 14, while this is a part of chromosome 14, and this is a part of chromosome 8, as a result of which derivative chromosome 8 and derivative chromosome 14 are produced. This part of chromosome 14, that codes for immunoglobulin C chain, but this part of chromosome 8 codes cellular myelocytomatosis. Close proximity of cellular myelocytomatosis to an enhancer. For the immunoglobulin C chain causes its overexpression in lymphoid cells. Cellular myelocytomatosis is a family of regulator genes and proto-oncogenes that code for transcription factors. As we have discussed, a translocation involves transposition of chromosome material, usually between chromosomes. 3 types are recognized: Centric Fusion, or Robertsonian, Reciprocal, and Insertional. Now let's discuss insertional translocations. Insertional translocation involves insertion of a deleted segment interstitially at another location. Let's understand it. This is chromosome 6, and this is chromosome 10 from chromosome 6. This segment is deleted, and at this part of chromosome 10, deleted segment of chromosome 10 is inserted. So, derivative chromosome 6 and derivative chromosome 10 are produced. At this point of derivative chromosome 10, deleted segment of chromosome 6 is inserted. It is extremely rare, and balanced carriers are usually healthy, but may produce chromosomally unbalanced offspring with either a duplication or a deletion. So, this was all about translocations. In our next video, we will talk about deletions, ring, and duplications in detail.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.